Okay. Hello, everyone. Yeah, I think now it's quite loud. Okay, today we have two very important events, two matches. One starts at 1, so I will keep on time. Yeah, and as I mentioned, in my first class was more fundamentals about virology and immunology. And today I will present what I do in the lab, what we do in the lab. And I will highlight the studies that we have been working in the last 3 years, 2 years and a half. So as I mentioned before, my lab works with immune response to emergent virus. So we study immunological response to viruses like dengue, chikungunya, Zika, SARS-CoV-2, and also monkeypox. But before getting started, I just want to let you know how I have turned my interest to the study of emergent virus. So I did my training in Brazil. I think I was introduced last time. And I did my training in Brazil. Is a country with multiple emergent virus and also endemic virus, like virus that are transmitted through arthropod vectors like dengue, Zika, chikungunya, Ropoche, Maiaro. And we can see there's multiple viruses that are endemic here in Brazil. All of these viruses, they have something in common, a feature in common, that is an RNA genome. So they can quickly spread into hosts and also adapt to different hosts and environmental change. And as someone that had interests in virology and immunology, I very natural, even during my training, I start focus on this kind of virus. And later on, I also work with viruses like HIV, and as I mentioned, SARS-CoV-2. And also more recently with monkeypox. Monkeypox is DNA virus, but all the others are RNA virus. So viruses are not only emerging in South America, so you can see here. So with West, Central Africa, South and Southeast Asia, together with South America, these places have the highest potential for viral disease spillover. And here we have a list of the infectious pathogens from the recent years. And you can see that all of them have something in common. They are RNA virus. So this virus, they can easily, as I mentioned before, they can easily adapt to host environmental conditions. And they can accumulate mutations. And we can have jumps into nature, especially if these mutations happen, for example, in antigenic sites that we talked last time, or in receptors related to antigenic recognition. So this is a timeline of pandemics. And here we can see that it is estimated that we have at least one new pathogen every year for the last 30 years. But as we can see here, the number of pathogens are increasing. So the ratio of these pathogens are increasing over time. So you can see that we can see more virus here. Of course, this timeline does not include only viral pathogens. There's also bacteria here. But recently, we can see that the infectious pathogens are especially RNA virus. And of course, here in 2020, we all know about SARS-CoV-2. And we could also include in these new years, for example, pathogens, as I mentioned before, like monkeypox. So the outline of this presentation, you will see that the outline of this presentation will follow very much the timeline of the pandemic. This was our previous 2 years and a half, and we are still having some waves of the pandemic. So back in March 2020, while the world was shutting down, our lab started working with SARS-CoV-2, as many other labs. And initially, we start with the characterization and identification of COVID-19 immunological markers. So after that, I will talk about a follow-up study that we focus more in antibody immune profiles and how that correlates with disease trajectory and disease outcome. And then we will talk also about emergency of new variants and also vaccine-induced immunity, different kinds of vaccines, homologous boosters, heterologous boosters. And we will also talk about the Omicron wave. I have a final part for this presentation, but I don't know, depending on how it goes. I have a project that is also not related to SARS-CoV-2, but it's related to therapy, to emergent virus. And if you have time, I will also go through that. But as I mentioned before, as you will see, this follows very much the timeline of the pandemic. And And as I mentioned, so back in March 2020, we start working with SARS-CoV-2. As soon as we have the first patient at Yale, we start doing a cohort of infected patients. Initially, we work mainly with the patients that were severe or moderate, but this is all hospitalized. So no asymptomatic, then we include more of these patients, but initially it was just severe patients. And our idea was try to understand how the immune response was associated with viral clearance, but how could also be associated with damage to the host. So this first cohort, this was multiple groups, multiple researchers, multiple doctors, nurses. I think everyone that experienced work with SARS-CoV-2 noticed that was a lot of people collaborating in it, a bunch of students, doctors, everyone together to work with this virus, especially at the beginning of the pandemic. And we enrolled this cohort. Initially, this cohort was composed in around 200 participants. As I mentioned, the majority of them were hospitalized patients. So you can see here that we stratify the patients based on levels of oxygen supplementation and also ICU admission. So we have at the base of the pyramid, we have negative controls that were healthcare workers. So the healthcare workers were screened every week. And if they were positive, of course, they then migrate. If they were severe or moderate patients, they were included in the cohort or they were not included anymore. But we have some negative controls, say PCR positive, serology negative at the base of the pyramid. Then we have patients that were hospitalized with low oxygen supplementation, moderate oxygen supplementation. And on the top of the pyramid, we have patients that were admitted to the ICU. And then we have patients that require mechanical ventilation. And unfortunately, at the top of the pyramid, patients that unfortunately die. And here we'll have like 0 to 6 will be the clinical score, and we will see this in some graphs. So for this cohort, we did more analysis related with PBMC's analysis. So we did collections of blood, and we did analysis of cells from the blood, and also plasma samples. So from the plasmas, we measure antibodies, but we also measure cytokines, chemokines, and viral loads. So this is very early in the pandemic. But as you can see here, this is an unsupervised heat map. And we did this unsupervised heat map based in classic immune cell types. So here you can see all the immune cell types. And we have, here you can see the different colors of our groups. So we have the healthcare workers in green, and we have our infected patients as moderate and severe. So you can see that this unsupervised cluster already stratify the participants infected or non-infected. So the highest intensity, it's more intense this cell type. So the darkest purple, we have more intensity of this cell. And we can see, for example, almost all patients that were non-infected, they have high levels of CD4s and CD8s, and they have low levels of neutrophils or eosinophils. And in contrast with that, we have the infected patients having high levels, especially of eosinophils, neutrophils, and low levels of CD4s and CD8s. So, of course, our group reported this, but many other groups report this decrease in T cells. So here is again, this is just a quantification. So here we have T cells, monocytes, neutrophils, and eosinophils. So in green, healthcare workers, and then infected participants, moderate and severe. And we definitely saw a reduction in T cells, but also we report an increase in other cell types like monocytes, eosinophils, and neutrophils. And we associated that with disease trajectory. Because this work was more focused on the cytokines and the inflammatory profile, we also quantify some cytokines. And here we can see increased levels of inflammatory cytokines. So for example, IL-6, but also interferons and IL-18, especially if we only compare infected participants compared to healthcare workers. So then we looked for the viral load. And we could see that the viral load, we could see a different trajectory already. So the viral load, this was a longitudinal analysis. So every patient was followed at least 7 times every 4 days. So here we can see the viral load. This is viral load on nasopharyngeal swabs. And this is a measurement of RNA. And here you can see the viral load over time. And we can see that their severe patients have higher viral load and compared to moderate patients that have a decrease in the viral load, and we couldn't see we could see persistent viral load in severe patients. They are not having a clearance so fast. And the viral load was driving the inflammatory cytokines, so people that could not clear the virus early on will have elevated levels of inflammatory cytokines, and this is a cycle, and inflammatory cytokines were associated, like interferons and TNF, with the viral load. So to try to more deeply understand the cytokines and the correlations with disease trajectory, we then did this unsupervised, the second unsupervised heat map. And here you can see it's very small, but this is all the cytokines we measured. And we separate the cytokines by different groups. So for example, here in the light purple, we have tissue repair factors. Then we have chemokines or lymphocytes associated with lymphocytes migration, type 2 and type 3 cytokines, and a mixed response. And what we could see here is that this unsupervised cluster already divides the patients in 3 clusters, cluster 1, 2, and 3. And if we look for cluster 1, it has more the signature, what we call signature A, that is related to grown tissue factors, and very low of the signatures that are related with this mix of cytokines. In the other hand, the cluster 3, we saw a lot of inflammatory cytokines and very low grown tissue and repair factors. So we tried to correlate this with disease trajectory. And here we can see the clinical scores that we saw at the pyramid, with clinical score of 0 being discharged from the hospital and 6 dead by COVID. And we could separate patients that falling on cluster 1, they have a better clinical prognosis, so they were discharged, while patients falling on cluster 2 and 3 were usually the patients that stay longer in the hospital and also have a worse disease prognosis. These clusters were also related with other complications like coagulopathies and also mortality. And we can see, for example, patients falling on cluster 3 have much higher complications and higher mortality when compared to patients falling on cluster 1.
Are these clusters the same division, severe, moderate, and the other are different?
No, these clusters was unsupervised. We just grabbed the cytokines and separate in any way. Of course, we define the number of clusters. It could be 3, it could be 4, it could be 5.
Is the same data set as the previous study?
Yes, this is exactly the same data set.
Okay. And do you have, how to say, can you compare the clusters and the previous categories?
Yes, we could compare. And definitely, again, these clusters were, definitely the cluster 3 included patients with more, especially the severe cases and the death cases, yes. And cluster 1, of course, here it's not healthcare workers. So here it's only included the hospitalized patients, okay? Not the controls. So to summarize this first study, so as I mentioned, I will just do highlights of each study. And the first study, what we saw is that patients with persistent viral load was detected in severe patients. The viral load was driving inflammatory cytokines. And we found a distinct cytokine profile in patients that will have severe disease or moderate severe. And we could distinguish these patients that will have different trajectories based on cytokines. So this paper was very cited. This was the first paper showing all the cytokines related with disease severity. So we really think that we helped dissecting this inflammatory phase of COVID. And what I'm not showing here, we also did a table showing some biomarkers that were related with severity and death that could be used in the clinic. High levels of IL-6, IL-18 were already used like very early on, before day 10, could be used as markers for distinguished patients that would develop severe or more moderate disease.
These markers, they can be observed after how many days of infection?
We did early markers, so until day 5, and then we did, but our more robust analysis was before day 10 because we have a higher number, so this could be done until day 10. And usually we define day 10 as a day very important for the disease trajectory. usually on day 10, the level of viral load, the level of inflammatory markers, but also other markers like D-dimer, I don't know if there's doctors in the room, but also the other markers, this could really be used as a prognosis for clinical trajectory.
Okay, and the level of the prediction, like how good it is?
The level of protection.
The prediction.
Oh, the prediction. I don't remember exactly. I think it was 95, but depending, we have the list and we have the levels, yes. So the second study was a follow-up of the initial study, and on this study we focused more on antibody response, and especially how antibodies correlate with disease trajectory. So here we extended our cohort, so we also include non-hospitalized, so asymptomatic and mild participants, and we not only stratify patients, as I mentioned before, by disease severity, but also by clinical outcome. So we separate in discharge and deceased patients. So the first thing we measure here was, previously I showed the viral load that was, severe patients have persistent viral load. Here is different. Here what we are measuring is the maximum viral load that each patient reached during the course of infection. So if a patient was in the hospital from day 3 to day 30, what was the peak of the viral load? And we compare that across different groups. And we compare in asymptomatic, moderate, and severe, and there's no difference. So the peak will be the same. The time that you clear the virus is different, but the peak is always the same. And so the peak is the same, but what we noticed that was different was the antibody levels. So the antibodies was always higher in who have more severe disease. So here we have controls. They're non-hospitalized, moderate, and severe, and we could this is IgM or IgG anti-spike, and we could see that the antibody levels correlate with disease severity. And what do we observe when we divided when we segregate these patients based on disease or discharge by outcome? What we saw here is the antibody levels over time. And we could see everyone reach the same antibody level. So here should be day 30. I'm sorry, it's lacking here the days from symptom onset, but here is around day 30 to day 40, and here this difference is around day 10. So what we see is like they reach same levels, but the timing that they reach the levels are different. So disease patients, they have a delay on mounting antibody response compared to discharged patients. So again, then we start doing analysis of neutralization. So neutralizing antibodies are antibodies that have a functional capacity, so they are able to block the virus to enter the cell. So these are functional antibodies. And here is a measurement of neutralizing antibodies by serum dilution. So in this assay, the viral load is always the same. The amount of virus that we add is always the same. What is changing is the antibodies, is the serum, the dilution of the serum. So we have 6 serum dilutions with a base 3. And so for this, so here the serum is more diluted, 30, 90. So we expect to 1 to 10, we will protect better than a serum that is more diluted because there's less antibodies. And what we can see here is that neutralization capacity is not different from disease and discharge in the peak of the response. But again, if we just look over time, we see, as we saw for antibody levels. So there's a delay on mounting neutralizing antibodies. So people that die, they don't mount antibody response very fast. So in the second week, they still don't have a lot of neutralizing antibodies. So because of that, then we stratify the patients based on neutralization capacity. So we have in pink what we call early neutralizers, and in green what is late neutralizers based on neutralization capacity on day 14. So on day 14, post-symptom onset, we have these 2 separations, and again the same graph by clinical score 0 being discharged from hospital, 6 is that by COVID. And people that do early neutralizing antibodies, they have a good prognosis, and they also have reduced mortality and reduced viral load compared with people that mount a late neutralizing antibody. So with this study, what we conclude is that the timing is very important. And not only, at this time we have, for example, studies from Eli Lilly that was in Regeneron that was producing monoclonal antibodies, and they already knew it that after day 10 or day 12, these antibodies would not work properly. They should be administered early on. So this was to show that the levels of antibodies per se are not different, but what is important is the timing of production.
Yeah, do you have an idea of why there's this different time to the production of the antibody?
Yes, we don't have a clear idea. Of course, there's multiple things that could be accounted.
Yeah.
All these analyses were adjusted for age, sex, all kind of parameters. But of course, we know that the majority of people that died with COVID were old individuals. And we know that neutralization capacity can be lower in the old individuals, but only that will not be able to explain. What I believe is happening is what we are working on right now. One of the things that we believe it's something that is called original antigenic sin. So original antigenic sin, it suggests that if you have an exposure for an antigen, in the second time, you will generate antibodies more related to that antigen than from the new antigen. And we believe that that happens more in old people. So we believe that old people still are mounting antibody response for common colds, and they don't change very fast for SARS-CoV-2 because this is very related epitopes. And we believe that is happening. There are already some studies in mouse models showing that. And that's also true for different variants. We also believe that that also happens for different variants.
So it would be like some dynamics of resource allocation and energy allocation in front of it, like you had a previous infection with some related epitope, and then you still producing this type of antibody, but the recombination process will just go normal, and you create the neutralizing antibody that you should to, but then you will not allocate the right amount of energy to produce more. Is that?
Yeah. And there's multiple things also going on. Of course, in older individuals, we have less TFH responses, we have less T cell responses. All of that together can result on this. And we also did measurements of TFH was reduced on this group, but we also believe like the antigenic scene is also having a major role, especially on older individuals here. So the second work, what we show is that elevated anti-S IgG levels were associated with worse disease outcome. And people that deceased COVID patients present a delay in virus-specific antibodies.
By the time we got vaccines, and so could the doctors and nurses use this information to I don't know, to try to fight this time independence of
no, this happened during, as I mentioned, for example, we understand why these neutralizing antibodies were not working very much later on. They should work early on. So if you work early on, you can reduce the viral load and you can reduce inflammation. So this was the most important, the biggest difference we saw after day 10 is definitely related with inflammation. So if you don't control very early on, you have a very high inflammation. And even if you produce then later on an antibody that is very high affinity to the virus and will control the virus, you have such cytokine storm and you have such inflammation that's the complication of the disease.
What is an anti-S IgG?
Anti-S is anti-spike. So we measure anti-spike and RBD. I will show because I think it's the next slide. This should be before, but anti-S is anti-spike, so it's in the envelope of the virus, and RBD is the interface of ACE2, and RBD is the site of connection virus and cell receptor. So what we show again is the clinical trajectory and outcome correlate with timing and not with the production of antibodies. And it's very important to not study only levels, and this is why longitudinal studies are so important. If we only collect the patient at one time point, we couldn't have seen this. So yes, so this is the spike. So the next follow-up that we have. So this study happened when we start being vaccinated in US. It was a little bit later in Brazil. But it was also at the same time that we start having the emergency of SARS-CoV-2 variants. So here we could see the variants of concern at the time. So we have alpha, beta, gamma, and delta, and also other variants of interest at the time. So like, for example, this 526, we have cows nearby. What's going on? Maybe it's the match, everyone's very excited, I don't know what's going on. So the 526 was a variant first identified in New York, was a variant of interest at the time. 427, 429, first described in California, also at the time declared as variants of interest. You can see some of the mutations and where are these mutations specifically here, but I will go also through that. And so at the time, we had main concerns about increasing transmissibility, increased severity, and vaccine escape or monoclonal antibody escape, therapy escape, because of this emergency of these variants. So yeah, so here I'm showing exactly the structure of the spike, because this study was, the goal of this study was to understand each specific mutation, what was the impact of each specific mutation on antibody response. So here is the virus itself, and here we can see the structure of the spike. So the spike is this homotrimers that projects from the surface of the virus. And so the spike can be divided in subunity 1 and subunity 2. The subunity 1, it's comprised the N-terminal domain, but also the receptor binding domain, or RBD. And the RBD is the one responsible for the interaction with ACE2, that is the cell receptor. And the subunity 2 also comprise other regions, like, for example, the transmembrane region and the fusion peptide. And basically the function of S2 is to fuse the membrane of the virus to the cell. Here we can see the 3D structure of the spike from its closed to open conformation. And here there's a furin cleavage site. So all SARS virus, they need a furin cleavage site. And this will expose different regions that will allow these bindings. And we also have the ACE2 binding site here represented in purple. So this is also to show that the RBD is the region that is immunodominant. So usually neutralizing antibodies will be against the RBD region. And we have 4 classes of neutralizing antibodies. Class 1, 2, 3 that binds to RBD. So here we can see the different classes. So we have class 1, for example, that it's ACE2 blocking antibodies, but that only binds in the open conformation. Then we have class 2 that blocks ACE2 in open and closed conformation. We have class 3 that does not bind to ACE2, but the antibodies bind in open and closed conformation. And the region 4, it's also, the binding is also outside ACE2, but only on open conformation. But this is just to say RBD is very important. We already know that all neutralizations, it's against that side. But we also have other regions of immunogenicity, as for example, the NTD or N-terminal domain here in purple, that can also some antibodies, neutralizing antibodies are related with to other sites. But definitely RBD will be a major site. So this study, as I mentioned, the idea first was to measure neutralizing response after vaccination, but also to measure the impact of each mutation, because we are seeing viruses with different kinds of mutations, and we wanted to know and also to predict what will be the next variants. If they have these mutations, they will be a concern. So we enroll a cohort of people that were vaccinated with mRNA vaccines. They received 2 doses of mRNA vaccines. And again, this is a longitudinal study. We collected individuals in 5 time points of collections prior to vaccination, but also 7 days post first dose, 28 days post second dose, 7, 28, and 70 days post second dose. Here you can see the different time points with the different colors. And we also separate the individuals based on the infection status. So people that were previously infected with the virus, and people that were not, we call naive. And these were the virus that we culture. So all the virus that we culture, we isolate from nasopharyngeal swabs from infected patients. So we receive samples from infected patients, we isolate the virus. All viruses that are here in black, we did the isolations. And this is just a comparison with other viruses that were public available at the time. And we have 18 different strains, and we selected these strains based on specific mutations. So all these strains, here we have the variants of concerns, alpha, beta, gamma, and delta, but also other strains. And here we can also see the names by the WHO that then change, alpha, beta, gamma, and delta, and today we have many more names than this. But this is the lineage. This is what we call lineage, and here is the name by the WHO. So this table looks complicated, but it's not as bad. So here it's all the strains that we have isolated, and here is the mutations, the site of the mutation. And here, so you can see that we isolated based on specific mutations. We tried all kind of combinations possible. And here is the neutralization levels. So here are the levels of neutralizing antibodies. And lineage A is the ancestral strain. So is the first SARS-CoV-2 virus. And all the others are mutations. And here, for example, you can see P1 is the gamma variant. You can see here in green is the delta. We can see some beta here. So to better understanding of this table, so the first thing, all viruses that we evaluate, in contrast with ancestral strain, they have this mutation on position 614 in the receptor binding motif. This mutation was demonstrated to not have impact in neutralization. Then all the mutations that we measure were located in the subunity 1 of the spike. So I show you the subunity 1 of the spike is the region that is related to the binding to the receptor. All of these mutations are concentrated in the subunity 1. So here, for example, we have mutation 18. That is the N-terminal domain. And for example, the mutation 1 is in the beta strain. We have one beta strain that has the mutation, the other doesn't. So we could address directly the impact of this mutation. Then we have deletion 69 and 70 also in the N-terminal domain. Then we have the mutations on the RBD. So 417, 452 are located in the epitopes of RBD, and all the others are located at RBD ACE2 interface. And then we have 681 located at the furin cleavage site that I show you was in red on the side. So the first thing that we could conclude when we did this analysis is that 12 of the 18 isolates that we did analysis reduced neutralization compared to ancestral strain. So you can see here the statistical difference for these strains. From the highest impact to the lowest impact, what we saw is that viruses all here in red that have these two mutations, 484 and 501, they reduce the neutralization the most. All the ones that are in red have these mutations, and this is true for alpha, this is true for beta, for example. And this was followed by viruses that have these mutations here in the 452, that was the delta. And we then did analysis to measure the impact of these mutations individually. So individually, we could see that almost all mutations have impact on neutralization, but the one that have the highest impact alone was the 452 that was found in the delta. But we also did this analysis, what about the mutations combined? And what we saw that combined, the virus with these two mutations have the highest impact on neutralization levels. And what we will see, so the main conclusions of this study was that this mutation that was located in the RBD, all these mutations that I mentioned, the ones that have the highest impact were in the RBD. They have the greatest impact, individual effect, but the combinations of 484 and 501 make the virus with the combinations with the highest concern for the vaccines. And we also did comparisons between people that were previously infected or not. I haven't showed this data. But this was true even today. If we look for the viruses that are emerging, they all have these mutations. They all have like the 484 and the 501. What we didn't expect that they will have much more. So then we have the Omicron emergency. Go ahead, I'm sorry.
Like in this experiment, he was made in vitro?
This was an in vitro experiment.
And which antibodies were used?
So this was plasma from the participants that were vaccinated. So what we have a cell line, and what we do a neutralization assay, we do the dilutions of the plasma from the patients that were in the different times of vaccination. We then add virus for 1 hour, and then the antibody binds to the virus, and then we add this to a cell culture. So if there was a binding of antibody and virus, the virus is not able to induce a plaque, so it's not able to induce cell death.
So the antibodies? So the antibodies, they were induced by the vaccine?
Yes.
So could be the case that the bigger neutralization was because of specificity of the antibodies instead of like the virus being better at evading the immune system?
Yeah, but we compare, so we have the controls. We also compare with patients that were not vaccinated, that were previously infected, and definitely people that were vaccinated have much higher levels of neutralizing antibodies. But again, we are comparing, so everyone was vaccinated, and the virus are able to escape more or less to the, so what suggests that these viruses are able to escape to antibodies that were produced that are specific for ancestral strain. Remember that just now we have vaccines that were that are available that are against, for example, Omicron, but all the other vaccines were against ancestral strain, and because of that, all our comparison was always on top of ancestral strain.
So this is conclusion. I thought the conclusion was that they were better at evading the immune system.
Yeah, no, the conclusion is that there's an escape of the antibody, yeah. So, okay, so here we have in November 2021, we start having the Omicron emerge, and this was a turning point in the pandemic. So the Omicron, on top of these variants of concerns, has multiple mutations, much more mutations, and up to 38 mutations compared to the other variants of concerns at the time. And in 2 months, Omicron was already like established in different countries and was the dominant variant across different countries. So at the time, again, we did the same kind of analysis. So we just, and as multiple researchers at the time, we just saw individuals that were vaccinated with mRNA vaccines, 2 doses, and we compared the VOCs at the time. So we have always comparison with ancestral, then alpha, delta, gamma, beta, and Omicron, and definitely we saw escape from Omicron. This was also reported by other groups, but what we did different on this manuscript, so at this time, this was January, so it was the peak of Omicron response. We did a collaboration with individuals from Dominican Republic, and like Brazil, Dominican Republic also used a lot of CoronaVac. So CoronaVac was a world most distributed vaccine at the beginning of the pandemic, and is an inactivated vaccine. And a lot of people were primed, what we call today primed, received 2 doses, fully vaccinated with CoronaVac. And at the time, a lot of papers start coming out about heterologous vaccination, so people that were, or 3 doses of mRNA vaccine, homologous but mRNA vaccines, but there was no studies showing, especially with inactivated vaccine, and especially if you did an heterologous vaccination. So this collaboration, what we did was individuals that received 2 doses of CoronaVac and 1 dose of Pfizer. And this was 101 participants, and we also collect before vaccination, before booster, and after booster. So they all have 2 doses of CoronaVac, and we did analysis like that, and also after the Pfizer vaccination booster. So here was the neutralization titers for ancestral, delta, and Omicron. And you can see just 2 doses of CoronaVac, there was no neutralization titers for Omicron in more than 100 participants.
So I'm just kind of confused with the setup of the study. You you just keep track of some participants of this study. You vaccinated them, and then collected before and after.
Yeah.
And I'm kind of surprised because people were always saying that how when one variant arrived, the other ones would be like consumed. But you have the ancestral there. The ancestral variant was still in the population at that time.
At the time, we were having, I think at the time here that we did this study, we still have Delta cases, but we definitely, if you remember last year, January, December, was a huge wave of Omicron and was dominant very fast. Yes, the study was individuals that were vaccinated with CoronaVac 2 doses, and then we collected blood and we did the analysis, and then they were vaccinated with Pfizer. And always our analysis were at 28 days after vaccination booster, because we saw that is the peak of the response for COVID vaccines. And all the studies we did the same time, so we can compare directly. But here, it's important also to note this here. So of course, post-booster, everyone, we can compare directly. It's 28 days. But here, before booster, people are in different times. There's people that received the vaccine 3 months ago, people that received 1 month ago. So that's why we have a lot of variation for the other strains. But there's no doubt there was no antibodies for Omicron, because it was 0 for everyone. And yeah, that was like, I don't know, that answer your question? And so what we saw is that after giving the booster with the Pfizer, these participants start producing, 85% of participants produce antibodies against Omicron. The levels were still low compared with ancestral and Delta, but definitely there was an increase in 80%. But yeah, then I'm already changing for, this is a new study. This study is still not published. This study was in collaboration with Brazil. And for this study is the same context. So we did analysis of people that were vaccinated with 2 doses, full vaccinated with CoronaVac. And then they received different boosters. So they received or Pfizer, or AstraZeneca, or CoronaVac again. So we have heterologous booster and homologous booster. And again, before vaccination and 28 days after vaccination, we did the analysis. So the idea of this study we started 1 year ago, especially because here in São Paulo, people were vaccinated with CoronaVac, and particularly older individuals were receiving homologous booster with CoronaVac. So the idea was to evaluate that and to understand if that was still enough, and especially now with the new Omicron wave. So in this study, we have already like BA.5, for example. And what we saw, so here, the gray, it's before vaccination, before booster, and the full color, it's after booster. And we have in red Pfizer, AstraZeneca in purple, and CoronaVac in blue. So here it's important to notice that this is just, again, IgG levels against S or RBD. And after booster, everyone increased. After the booster, there's an increase in antibody levels for heterologous or homologous, independent of the vaccine type. But definitely we can already see that the induced levels of CoronaVac were lower compared to the other vaccines, like 2 times increase compared to 5 times increase. But then we wanted to stratify participants by age, because what we know now, it's especially people that are vulnerable, like pregnant, older individuals, they are still more susceptible compared to younger individuals. So the idea here was to start doing this analysis by age. So this again is an unsupervised heat map, and here we have the different vaccine booster. So here the vaccine booster, it's here on top, so the different colors. So you can pay attention, like the blue is CoronaVac, and here we can also have the darkest is people that are age 50 years old or more, and the light color, it's less than 50 years. So this is not even elderly, it's just 50. And here what we could see, for example, this is just based on antibody levels, anti-spike and anti-RBD post-booster. And we can see already that participants that receive here, there's especially people that receive CoronaVac and AstraZeneca. And there's all kind of ages, and they have a very good response. What is the group 3 is the best, and these particular people that receive AstraZeneca. And the group here that it's completely blue, so this indicates less antibodies, it's especially people that receive CoronaVac. And the majority of people that are in this group are older individuals. So CoronaVac was inducing very low levels of antibodies, especially in older individuals. So this is the correlation of the vaccine type and by age, and it's very striking. So participants that receive a heterologous booster or Pfizer or AstraZeneca, independent of age, they reach the same levels of antibodies, but CoronaVac is related with age. So it's not related to vaccine type, it's related to vaccine type. It's not age-dependent deficiency of antibodies.
Sorry, why 5 clusters in the previous?
We just stratified. We did by 3. We did by 4 and by 5. We just use it in analysis, but the results, the clusters were almost the same.
This age and like difference in response of the antibodies is because of what? Like what is a system that is not working so well when you are young, or what?
Yeah, we don't know what it is. This is a very good question. We already knew it, that inactivated vaccine doesn't work very well in aged individuals. Like we also know that for flu. The efficacy of flu vaccine, inactivated one, works much better for young people than older individuals. But exactly why, we don't know. We are also trying to do that, but definitely we have these vaccines for so long and we still don't know exactly what is behind. Of course, to study mechanism in humans is very difficult, so definitely we are trying to address this also in mouse models, but we don't know. The only thing that we can conclude right now is that definitely the CoronaVac is not working very well for older individuals. This is just based on antibodies response, not neutralizing yet. So this is when we start doing analysis of neutralizings. What about neutralization capacity? Because again, we saw there was an increase after booster. It could be not very intense as is Pfizer, but that doesn't mean that it's not working. So then we start doing neutralizing to measure neutralization capacity. So here what we are seeing is this. So here different vaccines. We have Pfizer, AstraZeneca, CoronaVac, the different variants. So we have ancestral, we have Delta, BA1 that was responsible for the wave last year. Then we have BA2.12. BA2.12 was the first BA1 descendant that starts rising here in Brazil, but especially in the US and Europe. Then we have this XAF. XAF is a circulating recombinant, but it exists naturally. It's a combination of BA2 and BA1, but the spike is from BA2, so the neutralization tests will be against BA2. But this was also naturally happening. This was not in the lab that we did this recombination. And we also have here BA5. So the purple, the highest is the levels of neutralizing antibodies, the blue, its low levels, and here we have the total. This is the summary of everything. And if you just look here, so one more thing. So the outside, it's before booster, previously to the booster, and the inside is after the booster. Okay, so we can see that, for example, for ancestral, after the booster, we definitely have an increase in neutralizing antibodies. But if we look for BA5, then it starts getting more blue, or not ideal. And here we can see like the summary of everything. So you can see that definitely the CoronaVac is less purple. And here is the quantification of that. So here what we can see is that, so again, we are dividing by age. So here we have people that are over 50 or less than 50 years old. And here we have Pfizer, AstraZeneca, and CoronaVac. And in gray, before booster, in full color after booster, and again the different variants. So we could see that coronavac, for example, and all the vaccines induced an increase for ancestral and for Delta. But if you check, especially for older individuals, especially this here, coronavac before or after booster, for any of the Omicron sublineage that we have tested, there's no difference. So there's no difference before or after booster.
I don't know if I missed it, but could you briefly explain how do you do this neutralization assay?
Yes, so the neutralization assay, what we do, to have a neutralization assay, the first thing you need is a cell that dies from infection. So we use cells from monkeys, it's kidney epithelial cells. We have been using these cells during the entire pandemic, and these cells, when there's a viral infection, the virus enters the cells and induces cell death. And because of that, instead of seeing a well that it's completely covered with cells, you see a dot because the cell is dead, so the cell is not there anymore. And what we do dilutions of the serum from the patients. So we receive patients' plasma, and we do different dilutions to dilute the antibodies. And we use the same amount of the virus for all dilutions. Then we get these dilutions and we add to the cells. And then we have 6 points of dilutions and we do a curve. So with that curve, we can generate this PRNT50. So it's 50% of plaque reduction. So if we see more plaques, it's because there's more cell death. If we see that the cells, there's no plaques, it's because there's neutralizing antibodies. Because we do this incubation of antibodies and virus, and if the antibody blocks the virus, when we add to the cell, the virus cannot enter because it was blocked. I'm sorry, it's quite complicated. I should have a figure of this, but
That graph you're referring to is the one that was at the beginning, that was like blue and purple?
yes. Yeah, we always use exactly the same colors. And yeah, here is just to show that even for younger individuals, we can see that already it's not working very well. And again, to be very clear, coronavac saved many lives at the beginning of the pandemic. All vaccines were very important. All the vaccines reduce hospitalization, reduce cell death. What we're saying right now, the vaccines need to keep up the pace of the new variants. And what we are seeing is that for vulnerable patients, like individuals, like people that are more than 50 years old, coronavac has no effect anymore against Omicron sublineage. It worked well for ancestral, it worked well for Delta, but it's not working for Omicron sublineage in these patients.
This lineage of cells were engineered to express the ACE2 receptor.
The cells, say again, I'm sorry, it's just...
They were engineered to express the ACE2 receptor.
They were. So these cells were engineered to have, they have what we call tempers. So they have, yeah. So these cells need to have, like for example, ACE2 or tempers that are co-receptors. And therefore they were engineered to have these receptors. And we always check the levels of the receptors because some cell passage, the receptors start decreasing. So we can just use the cell lines for like 5 to 10 passage.
Okay, and why not use some lung epithelial cells?
Oh, we used a bunch of, we use lung cells, we use a bunch of cells. It's just because usually people use pseudovirus. Yeah. We work with the virus itself. This is all BSL-3. This takes very long, but we really wanted to do with the virus itself, the wild-type virus. But we test other cells. The problem is other cells takes like 6 days to grow. Oh, some cells don't die, other cells die. So this is easy to quantify. The other way of doing could be, for example, the virus expressing GFP. So you just go to the facts and just see the GFP population. We can also do that, yeah. There's multiple ways of doing, but we test in other cells. Microphone.
Hi. I remember during pandemics a discussion about levels of antibody production after coronavac, immunization, and everything. And I remember some people saying that it's true that antibody response is not the only response that we have in an immunity response. Did you did you do, or someone did evaluation on cellular response
Yeah. So
after
yeah, that's very
coronavirus
important. And again, I'm just showing neutralization results. Definitely we know that neutralizing antibodies are extremely important. The vaccines were designed based on neutralizing antibodies. But definitely other cell types like T cell response are also important to the control of infection. And again, this data that I'm showing not necessarily say, oh, the person that has 0 neutralizing antibodies will die from infection, will be hospitalized. I'm not 100% sure about that. Of course, we have relation that suggests that's not ideal. But we have the cells. We haven't done yet the analysis of these cells, but we have the PBMCs, we have the cells. We did for the other studies, but not yet for this one. And what we know, we also have a collaboration with other researchers that did epidemiological studies in Brazil and also compare people that were received coronavac during this Omicron wave infected with BA.5, how they were responding. Are they being more hospitalized compared to the other vaccines? And that's true. So they compare also AstraZeneca, they compare also Pfizer, and people, older individuals that are receiving coronavac having higher levels of hospitalizations compared to other groups of vaccines. This is in collaboration with Albert Koch, Julio Croda, I don't know, some of you may know him. Yeah. So yeah, so with this study, what we concluded is definitely that homologous regimes induce low antibody response, and there's a remarkable impact in older individuals. Lower antibody response is vaccine specific rather than being associated with age. It's not just related with age. The vaccine will induce different responses. And the homologous coronavac boosters do not improve neutralizing response against Omicron sublineage in older adults. And what we are suggesting is that individuals that receive 2 doses of coronavac or even like 3 doses of coronavac should be vaccinated with another vaccine. What time it is? we still have time. Okay. So yeah, so this is about this study. This is what we did right now. The other thing that I have here just to show you, just for curiosity, I don't know, because we are having a bunch of new variants circulating right now. So of course this study we still did with BA.5, but we have, and here you can see the specific mutations comparing, for example, BA.5 with BA.1. Of course, these the Omicron sublineage, they are all related. They share mutations, but they have individual mutations. And even if you remember, for example, BA.4 and BA.5, they acquired this 452. I don't know if you remember, but this was the one mutation that was with Delta that we showed before. And these mutations that they are accumulating, especially in the RBD-ACE2 interface. And even now, we are seeing here, I think we are seeing BXS. BXX is a recombinant circulant variant of BA12 with BA2.75, so it's a new one. But I think the one that everyone is talking about now is the BQ1 and the BQ1.1. BQ1 and BQ1.1 are sublineage of BA5, so they have the same mutations that are present in BA5, but they have on top additional mutations, and they are also concentrated on these regions here. So because of that, some manuscripts are showing some escape from antibody responses. And I think it was approved recently, also the bivalent here in Brazil. I don't know if it's already available. I don't think so, but it should be available soon. At least in January, maybe that changed. And so the last part of this presentation, I don't know if you have any questions about COVID.
So if I understand correctly, before COVID there was another epidemic of a similar virus in China, right? Or in some
No, what we have before...
I think SARS. Something.
Oh yeah, it's SARS you're talking about. Yeah, we have two different things. We have common cold virus that were there all the time, that we call common cold. And we also have the SARS one and MERS. So SARS-1 was also related with SARS-CoV-2, and it happened before. It also started in China, but we also have some cases in the US, but it was quickly contained. And we also have MERS. MERS was much more lethal. It's also related to SARS. It was much more lethal, but we have much less cases, and it used a different receptor. It's not ACE2.
Okay, and is the mechanism of these diseases similar in the sense of like, well, at least insofar as lethality for humans goes, it is related to an inflammatory response that it generates?
Yes, SARS-1 and SARS-2 are very related. They share very similarities in the genome, the immune response. The infection, the cell types that they infected are a little bit different. And yeah, it was but they are extremely related, and also the induction of antibodies. There are studies showing antibodies for SARS-CoV-2 could also cross protect for SARS-1, for example. Yes, they are extremely related.
And I don't know. I'm trying to formulate a question here, but I'm coming from a place of strong ignorance about a lot of things. So I don't know if this makes sense, but the selection pressure for these viruses as they propagate through, let's say, animal populations, humans, and so on, is it mostly related to escaping the immune system, or is there also selection pressure for, let's say, attenuating the immune response? Because because if the virus itself is not increasing its fitness by having the organism, the host, die from an immune response, right? Of an inflammatory response.
Yeah.
So is there any selection pressure for to attenuate the immune response of the host?
So mutations are random. Mutations will happen in the random way, right? We will have all kinds of mutations. But of course, we will have, like, for example, what we saw, for example, in Brazil, we have the P1 or the gamma, and in South Africa, we have the beta. And these mutations happen in places that in South Africa was a place that we were having a massive vaccination, and in Brazil was in a place that we have a lot of people infected. So definitely that suggested that these mutations came from a pressure from people that were previously infected and have a good antibody response. So definitely the immune response will induce some pressure for, and this is the assays that we also do in the lab, and this was assays, for example, from Michel de Saint-Vai and Paul Beniache that were very sophisticated very early on in the pandemic. What they did is, like, you develop monoclonal antibodies or plasma from, we can select the best individuals with a higher neutralization capacity. We get plasma from that in the cell types, and they did that assay. We have that antibodies, we have a bunch of monoclonal antibodies, and we add in the cell culture, and we start collecting, and we passage that in the cells, the virus with these antibodies in the cells. What they saw in advance, they saw all the mutations that we later on saw that were going to happen, they saw already in this assay. So they knew that the mutations were converging for some regions. They saw that, and this was exactly the mutations that would happen in the future. So definitely the immune system induced pressure. Of course, there are other things. Of course, there are viruses that we pass in animals, and they will start adapting. There's mouse-adapted strains. And of course, again, these mutations will be random, but the immune system, evasion of interferon response, evasion of T-cell response, the gamma variant is one that is associated with the evasion of MHC class 1. There was a mutation in a specific region that was not observed before. So definitely the immune system will induce pressure against different kinds of virus that will be able to still propagate in nature. And of course, this is why it's so important to have vaccinations happening at the same time. If you have a pool of individuals that are vaccinations, others that are not, the virus will gain space to advance, let's say. It's not correct to say that. Evolutionists, please correct me. But yeah, but definitely the immune system will induce pressure. And we saw that in multiple ways. We saw that in real life, we saw that in the lab. So the last part of this class is about the development of new therapeutics. So one part of the lab, we definitely study very basic immune response to various infections, but the idea is also to generate therapeutics. So this idea came from a previous study. That this study, what we did, we infect mice with, so first of all, the idea of this is also to induce, I work with therapeutics for emergent virus. But the idea of this came from a previous study from the lab that I did my postdoc. So after a herpes infection, if we infect mice with herpes, the mice will induce, and this is not a little infection, mice will have a memory response. They will generate a memory pool. This memory pool in a secondary infection will migrate for the CNS. Here is DRG, dorsal root ganglia. And these cells, specific CD4 cells, they start producing interferon, and they change the permeability of these regions, and they allow antibodies to cross to the CNS and to prevent the viral infection. So this was the first observation for this study. And then what I wanted to see, if I could do this with a virus that infects the brain, and if we could translate these mechanisms that is induced by virus as a protection. So my idea first was to translate this to the brain. So we saw in the DRG, but we wanted to see in the brain. So what we do is immunize mice with herpes, and we wait 5 weeks to develop a memory response. So they will develop CD4, CD8, B cells. And then we do a stimulation just with a peptide protein intranasally. So we just use a peptide that is specific for CD4s, and we do intranasal. So when we do, this peptide is very small, can cross the BBB, the brain barrier, and then this peptide recruits CD4s, and the idea is that the CD4s will produce interferon, and interferon can modulate permeability of the brain and allow antibodies to have access. So we did this kind of analysis, and what we saw, so here the dotted line is the control, and here was the mice that were immunized and treated with the peptide. And what we could see is that we can induce brain permeability on day 4 and day 6, and that close on day 10. So it's a very safe way. We have a window to open the BBB and then it closes. So during this window, we thought if we inject something in the circulation, this will go to the brain because the BBB is open. So we can give access to treatments for brain cancer or treatments to reach the brain that were not available before. So this is just to show that it works. So first thing, this is just the peptide, and this is the entire head. So this is the eye, this is the olfactory bulb, so the brain, and here is the nose. And here is just to show if we do the peptide is labeled, the peptide is in the nose, it will be in the nose. And then, but also goes to the brain. So we can start seeing the peptide also in the olfactory bulb. And here is the same thing. So this is just a microscopy showing animals that were immunized and treated with the peptide. This is olfactory bulb, so very at the beginning of the brain. And the peptide can go to the brain. The next thing we could see is that, so here is not the peptide anymore. So here is a measurement of brain permeability. So this is dextran. And this is the DRG again. So we are giving peptides in the nose, and we are seeing if we can even open the DRG. So here it's the animal that was not immunized, and here is the animal that was immunized and received the peptides. And you can see that even like that, it opens the DRG, so there's permeability. So here is a measurement of IgG, as I mentioned. So what we did is, when we have this opening, we treat the mice with antibodies in the circulation, and we measure if the antibodies are going to the brain. So usually antibodies are too big, they don't cross. So that's the difficulty for viral infections or even for other therapeutics to reach the brain. They don't have access. And what we saw is with this strategy, we can allow antibodies access to the brain on day 6. And this close, what is very important, because if it was open, it will not be good. So it needs to close, it needs to be safe, and it's closing. And here is also, instead of labeling the peptides, here what I'm labeling is the antibodies. So the antibodies are going to the brain. So here is when we treat it with the peptides that are specific for CD4s, and here when we treat it with unrelated peptides. So the antibodies are going to the brain. So here the idea was to check if this would work, for example, for cancer models. So again, we immunized animals with herpes, non-lethal dose. We wait to the development of memory response. Then we introduce the cancer. So there's a tumor that is intracranially implanted. And then we treat the mice with the peptides that are specific for herpes T cells. And then we give PD-1. So again, PD-1 does not have access to the brain usually. And this is a glioblastoma model. So there's no treatment. And because there's no access.
I just lost one part. That is why the herpes is important in the...
Because we need, what is opening the BBB? It's T cells that are specific